guest tonight. We have Wes Hutchins. Wes Hutchins is currently an undergrad student. He's a sophomore at University of Maine, Orono, where he's been working on this monarch project that he's going to talk about this evening. So he'll talk all about that. He's working with Dr. Amber Roth up there. His major currently is wildlife ecology, and he has a passion for wildlife of all varieties, with a special focus on insects, of course, and that's exactly why we have Wes here. In addition to that short intro, I've also already gone ahead and posted a link in the chat box. So if folks can go ahead and open that, you can read that at your leisure. So Wes was also nice enough to provide us with a blog post for the website. So that's available, probably best after Wes is done talking, so you can give him our full and undivided attention. So that's it for me. So again, just a reminder to folks, please stay muted and keep your webcams off. Otherwise, I'll go ahead and do that for you if you happen to unmute yourselves. But without further ado, I will turn it over to Wes. Thanks very much.
All right, thanks for that, Mike. All right, guys. So thank you everybody for being here and giving me your evening here. So like Mike said, my name is Wesley Hutchins, and this presentation is going to be about my experience with this with my project that involves tagging monarchs at the Fields Pond Audubon Center. So hold on one second. Oh, I can't, hold on. Minor technical difficulty. Okay, there we go. Okay, yeah, my screen was frozen for a second. I couldn't move my slides. So yeah, before we get too deep into it, here's just a brief introduction about myself. Some of this Mike just said just now, but my name is Wesley Hutchins. I'm a sophomore at the University of Maine at Orono. My major is wildlife ecology. And this has been something that wildlife ecology is something that I've always known I wanted to pursue. I've always been fascinated with wildlife of all shapes and sizes. This is me, I think 2 years ago. With the third or fourth snapping turtle that I've ever caught. So, I've always been, the type to, enjoy playing outside. Even today as a 19-year-old, people ask me what I like to do, and I say, I play outside, so wildlife ecology is something that I've wanted to study my entire life. And I knew from a very young age that this is what I wanted to really have a career focused on one day. So yeah, there's never any confusion for me that this was unlike what I wanted to do. I always knew I wanted to do something wildlife related. So here's a little bit more about me. That's me on the right when I was a wee lad, probably about 8 or 9 years old. And I'm releasing one of the many monarch butterflies that I have raised across the course of my life. I have been raising monarchs basically since I could walk. I've been doing this since I was a very small kid. So, I've been collecting the caterpillars, bringing them inside and, watching them grow up for literally my entire life. So I have quite literally a lifetime of experience handling and interacting with all stages of the monarch throughout its life cycle. And I've been to the Fields Pond Audubon Center many times as a child, just for fun, just hiking around in the woods and looking for bugs and birds in the fields and all the trails they have. And, and all this just sort of helped to, further, make stronger my, love and passion for wildlife and just the outdoors in general, And then when I was about 14, I actually led some field trips for elementary school kids at the Audubon Center. So I would just, walk them on the trails and, point out the different kinds of trees and the, shift in habitat that you get as you sort of hike up that hill that they have and, point out the bugs and the snakes and all those things, And one thing that I learned from that experience was that, first of all, I love being outside. And second of all, I don't really love teaching this to other people. So I learned that this isn't really for me. So then I was like, well, bugs are really cool. And so the insects are really cool and I'm really passionate about them. I don't love sort of, teaching little kids about them, but bugs are pretty cool. So I dug around a little bit deeper and I discovered the Maine Entomological Society and joined that probably when I was 15 or 16. So I've been a member for a few years now. So here's a little bit more about me. I do have some entomological related experience before coming to college. I would volunteer at the Maine State Museum storage facility in Augusta. So basically they have the Maine State Museum has a large insect collection that goes on behind the scenes that needs curating every now and then. Stuff needs to be sorted and labeled occasionally. I once spent a good deal of time sorting through their spider collection and just literally sorting them into different boxes and where they belonged and all that stuff. So these two pictures I have on here are pictures of some of the specimens behind the scenes at the museum. I don't have any pictures of me actually working with insects, but they got a polar bear back there and they got the Eskimo curlew and some passenger pigeons back there. So it's just really cool. I didn't work with the taxidermy very much, but if I had a picture of me with the insects, that's what would be here. But I figured this was the next best thing. So in high school, I applied for and was awarded a grant from the International Coleoptera Society to conduct research on beetles in Swanville, Maine, which is where I'm from. So I've lived in Swanville, Maine my entire life, and I was able to conduct essentially a survey on what insect families were most abundant in the woods behind my house. And that was when I was a senior in high school. So I have had experience collecting insects and like labeling and sorting and dealing with insect specimens for quite a long time. Let's see. Yes, so these are my college beginnings. So I was accepted to the University of Maine as a top scholar award winner. So basically, a UMaine top scholar is are the top 20 applicants, and they receive $500 a year to be put toward undergraduate research. So as I was investigating opportunities for undergraduate research, I found Dr. Amber Roth from the forestry department and her work with tracking birds and butterflies with low-tech transmitter tags. And we'll get more into detail on what exactly these low-tech transmitter tags are a little later. And this project was mostly focused on birds, but there was a portion of the project that involved tagging monarch butterflies. And I was basically, I reached out to Dr. Roth and, we emailed back and forth about her project. And I was basically put in charge of the monarch portion of this project. And I came up with really one main research question, and that was, how long do monarchs stay in the area where they lived as caterpillars? And as I was studying this question, sort of another thing that would be cool to see would be like, what trajectory do the monarchs take as they migrate away from Maine? But really my main research question was, how long do they stay in the area where they lived as caterpillars? And this butterfly tracking project and the bird tracking project both were taking place at the Fields Pond Audubon Center in Holden. So let's actually start talking about the monarch tagging project itself. So this picture we have right here is one of the tags that I was affixing to my monarch butterflies. Each tag weighs about 1/10 of a gram, 0.13 grams. And it's really just like a tiny white pill with a hair-like antenna running out the back of it. And now that antenna is about, I'd say 3 to 4 inches long. It's about 4 inches long. And it lasts about a month before it dies. 28 days is the guaranteed minimum. It's possible that it could last a few days longer than that, but the guaranteed minimum battery life of one of these things is 28 days. And so with the $500 from my top scholarship and a very generous grant from the Maine Entomological Society itself, I was able to purchase 5 tags, 5 of these little nanotags from the company Low-Tech. And so how do these tags work? Once they're on an animal, how do they work? And we'll get into exactly the physical actual tagging process a little later. But right now, how do these tags work? So on this top image, we have what we call a Motus tower. And the Motus tower runs on what is called the Motus Wildlife Tracking System, which is this like online database that picks up signals from registered tags whenever a registered tag flies within 8 miles of a MODIS tower antenna, like this one. So this is the MODIS antenna that you can find at the Audubon Center. You know, the top image is just sort of a closer picture, and the bottom picture is the building at the Audubon Center, and you can see the tower sticking up behind it. So before you can use a MODIS tower, you have to register your tags with the MODIS wildlife tracking system itself before you can actually see the data on the MODIS website that is collected through this tower. And that's the cool thing about MODIS, is that all the data collected from MODIS towers can be seen by anyone on their website. So all you gotta do is go to modis.org and you can see the data from all sorts of projects that people are doing across the country that involve this technology. So, this is a map, before going on, this is a map of MODIS towers sort of in the eastern US and Canada. So like the yellow dot beneath the N in Maine, that's the one, that's the MODIS antenna at MODIS antenna tower at Fields Pond. Looks like the closest one is, I don't know, maybe that's in Bangor somewhere, right on the N. A bunch over here in Nova Scotia, and then a bunch over here in Ontario. So yeah, there's a lot. And the idea is that you can, an animal that is wearing a registered tag, whenever it flies in proximity to one of these towers, then you will be able to find out ultimately which tower the animal passed by, so you can get an idea of where exactly it traveled across the country. But we'll get a little bit more into that later. So, how does one activate a tag with MODIS so that you can see the tag's data on the MODIS website? So basically, there were 3 things that were needed to activate and then register the tags. And those were the tag activator, a FunCube dongle, and Audacity software on my laptop. So what are each of those things? The tag activator is this black and green box in the upper picture here. It's just a plastic box with a hole cut in it. And you just lower the tag itself into the hole, like you can see in this picture. And then you hold down on a button on the tag activator itself. And this action enables the tag to actually be activated. I'm not sure like what mechanics are actually going on within the box. There's like some flashing light that I can see, you know, coming out of this hole. But that's how you activate the tag. You lower the tag into the hole, press the activate button, and then the tag is now on. And then if you were to turn the tag off, you would stick it back in the hole and then press the off button, and that turns the tag off. So the FunCube dongle, which is, I don't know who named that, it's totally ridiculous. But the FunCube dongle is this USB looking thing with an antenna attached to it in the picture on the bottom here that's plugged into my laptop. And basically what this does, this is basically just the antenna that listens for the activated tag, and then it displays the pings from the tag on my laptop through a piece of software called Audacity. So once each tag is on, every 30 seconds, it releases a little ping, releases a little, like, it sounds like a little cricket almost. It's like a little cricketing pinging sound. So once the tag is activated, its sound is picked up by the FunCube dongle antenna thing. And then the ping is actually shown on the Audacity screen, is shown on the screen through Audacity. So you can see that it is on, and you can hear that it's on. It actually will make a physical sound through the laptop speakers. And the Audacity software is also what was used to record each tag's unique sound. So basically each tag sounds and looks superficially the same, but each one of them is in fact actually unique from the other ones ever so slightly. So in order to register these tags with MODIS, a recording of each tag has to be created and then like submitted away to MODIS. And this basically tells the MODIS wildlife tracking system, okay, this is what the tag sounds like. This is what it's going to tell the MODIS antenna towers to listen for, to be on the lookout for. So this is the process how to activate a tag. And then after activating them, all of the tags were deactivated until they were actually ready for use, so that we wouldn't waste that battery life. you know, like as I mentioned before, you know, we only have about 28 days of battery life. So, you know, every minute is precious. So, you know, got to conserve the battery life. Also, one thing that I forgot to mention sort of near the beginning that I should have brought up near the beginning is that there will be a Q&A near the end. So I'll just take all your questions at the end if you have any questions or anything. That's sort of unrelated to anything going on right now, just so y'all know. Okay, so let's take a break from looking at the technology and actually start thinking about the caterpillars themselves. So all of the caterpillars in this project were collected as caterpillars from the grounds of the Fields Pond Audubon Center. So I had permission from David Lehman, who works at the Audubon Center, to collect monarch caterpillars from the grounds. Normally, you're not allowed to collect anything from nature at any Audubon Center. You know, it's sort of like a nature preserve, but I was given permission to collect caterpillars for this project. And each caterpillar was collected and brought indoors and raised indoors until they emerged as adults. And I collected many, many more caterpillars than I actually needed. You know, like I said before, I only was able to buy 5 of these little tags. So, but I collected way more than 5 monarchs. I think I ended up collecting probably a total of 25 or 30. So I had a lot, and all of the extra adults were just released at Fields Pond. So like, I still brought them back to the place where I collected them, even though they weren't even though not all of them are going to be used for the project. So yeah, here in this upper picture, just a couple of the caterpillars I had, you know, just chilling, eating the leaves in this mesh tent that I had for them. And then there's a chrysalis down here. You know, we're all familiar with the monarch butterfly. You know, we all know what these caterpillars look like, what the chrysalis looks like. You know, it's a charismatic insect species. Okay, so basically, not all monarchs are worthy to carry a low-tech tag, because each tag weighs about 1/10 of a gram, which, you know, obviously is very tiny, but to a monarch, it's still small, but less tiny. So basically, each monarch must weigh at least 1/2 gram to be able to carry a tag. So no monarch that weighed, you know, 0.5 grams or less was fitted with a tag. These monarchs were released outside without being tagged. So now you're all thinking to yourselves, how does one weigh a monarch butterfly? And the short answer is really, really carefully. So basically what you do is, or what I did was I used a sensitive kitchen scale, and I would weigh an empty envelope and write down that number. And then I would place the monarch butterfly into this envelope and then weigh them both. And then I would just do algebra, and I would subtract the weight of the empty envelope from the weight of the envelope with the monarch inside. And that's how you'd get the weight of just the monarch. So the monarch was only ever in the envelope for like, you know, 2 or 3 seconds max. So, you know, I wasn't keeping them in there for too long, you know, stressing them out. So don't worry about that. So how to tag a butterfly? Short answer again, very, very carefully. You know, butterflies are fragile creatures. You know, they're not as fragile as a lot of people think. You know, they are out in nature, you know, where it winds and rains and all that, you know. So they're not as fragile as you think, but they still, you know, are quite breakable. So, you know, you do have to be very careful and take it very slow when you're actually affixing a tag to a butterfly. So first things first, before I went about tagging the monarch, was I would actually activate the tag, you know, with the tag activator box. I'd lower it in the hole, press the button, make sure it's activated. I could see on my laptop screen with the Audacity software open that the tag is pinging and sending up little spikes. So I could see that the tag is on, and it was ready to go. So now it was time to actually stick the tags on the monarchs themselves. So basically, each tag was affixed to the underside of the monarch's abdomen, close to the junction of the abdomen and the thorax. So that position would keep the tag out of the way of the legs and the wings and the spiracles. The spiracles being the holes that run along the sides of the abdomens through which the insect breathes. So the tag was out of the way of all that stuff. So basically what I would do is I would pick up the monarch with my thumb and middle finger, and then I would use my pointing finger to sort of hold the monarch's hand. The monarch would be very willing to like just have something to cling to with its hands, with its legs while I was doing this. So I basically hold it belly up with my thumb and middle finger, use my pointer finger to sort of keep its legs out of the way. And then with my other hand, I would pick up the tag, and I would dip it in a tiny like infinitesimally small puddle of just crazy glue. And then I would hold the crazy glue onto the underside of the abdomen of the monarch for about 15 seconds. And then I would let go of the tag, and then I would put the monarch back in the enclosure and then let go of it. And that was it. It's pretty simple, you know? And then every tagged monarch was held indoors overnight in this giant mesh pop-up tent thing that I bought online, to give them time to adjust to the weight of the tag. So I didn't, I didn't want to just like fling them outside in nature with this tag on them and just expect them to adjust, out in the wild, so it was good for them to have a little bit of time to practice flying around a little bit with this little bit of added weight on their bodies before putting them back out into the wild. Yeah, and it was cool to see them sort of in this mesh tent that I bought for them, sort of flying around. They would fly from one side of the wall to the other, so it really doesn't weigh that much, and it didn't hinder their flight. It really didn't hinder that flight at all, basically. So here's a picture of here's a couple pictures of monarchs with tags on them. So there's one on the left, sorry, the mesh is in the way. You can actually see the white pill there affixed to the underside of the monarch, and you can see the long hair-like antenna running off the back of it. And on this picture on the right, you can see the monarch from the top. You can't see the actual tag itself, but you can see this long antenna running off behind it. About 4 inches long. So before releasing the monarch, yep, as I said before, each tagged individual was held overnight indoors to acclimate to the tag. And during this time, I offered them some squished up fruit, some red Gatorade, and I mixed up some honey with some water, and I'd put that on a sponge and offer that to them. And then I would use a toothpick to actually like unroll their proboscis and dip it into these food sources so that they knew it was there. And monarchs actually can taste through their feet to some extent. So whenever I would put one in the enclosure, I would just place it on the food and then introduce its proboscis to it. So they knew it was there with both their feet and their proboscis, and they were able to come and take a drink of any of these things if they wanted. I offered all 3 of these at the same time. Honey water seemed to be their favorite, but I just kept offering all 3 of them just in case. And I had nail polish remover sort of on the side in case I ever needed to remove any of the tags. I never had to use any, but I had it on hand just in case, like a monarch's leg got stuck under or something went awry with the glue and I had to like remove a tag from the animal. Nail polish remover undoes the crazy glue, but I never had to use it, thankfully. So after being kept overnight in the mesh tent, if the butterflies are still flying around and active strong, which all five of mine were, then the time came to release them at Fields Pond. So all of the monarchs were released at Fields Pond where they were collected. 3 of the 5 tagged individuals flew out of sight immediately. Like you wouldn't even have known that they had a tag on them. They just flew straight up and away. And then 2 of the 5 individuals took their time a little bit with it. They landed on nearby objects and just sort of hung there for a few minutes. And then I would come back and check on them every few minutes until they left. And as I mentioned before, all of the extra adults that didn't end up being tagged also were released at Fields Pond. And you can see in this picture on the right, that's me holding one of the monarchs. You can see the antenna running off the back of it. And this is me like just seconds before releasing it. There were some field trips of elementary school kids at the Audubon Center on some of the days that I was releasing some of my monarchs. So that was kind of cool, actually, to be able to sort of tell them what I was doing and sort of explain to them what was going on. It kind of brought me back to the days when I used to do this, when I used to lead field trips at the Audubon Center. All 5 of the monarchs did get named by the kids. I believe there were kids there every time I released them, actually. So all 5 of them were named by the kids before they went off. I believe we had, I don't think I even remember all of them. One of them was Billy II, and there was no Billy I. That's the one that I remember. So this might be, I think this is Billy II in my hand right here. So yeah, that was fun. That was fun to see the kids, watch the kids name them. That was funny. So now that the monarchs were out in nature, I was able to track their approximate movements on my laptop by looking at the Modus website. So, oh, sorry. Wait, what did I do? Hold on. Nope, nope, nope. Hold on. I just totally gave away the ending of my whole presentation. Sorry about that. Let me see here. There we go. All right, sorry. I don't know what happened there. Anyway, like I mentioned before, the burst rate of each tag was approximately 30 seconds, about 28 seconds to be even more specific. That means every 30 seconds, it sends out a little ping that a Modus antenna tower can hear if the tag is within range. And the Modus tower uploads all of that data, all the data that it gets in a day, to the Modus website. So this picture on the right here is just a screenshot from the Modus website. And you can see each one of the rows of this table is a ping from a tag that was within range of the Fields Pond Modus tower. So there are some birds on here. You know, I see Swainson's thrush and bobolinks on here. These are some other birds that have been affixed with the similar tracking devices to the Monarchs, other people's projects. And there are some Monarchs. These Monarchs are mine. These are the Monarchs that I tag. And you can see, like, you know, on August 28th, 2021, a Monarch was picked up, you know, and you can see that. And this table is much bigger. This is, there's like, I think 4 or 5 pages of this table on the Modus website. So this table is obviously a lot bigger than this. So while the Modus website does show a lot of the data that is sent in by the tags, it doesn't show all of it. So because the burst rate of the tag was 30 seconds, like at the end of the day, that's a lot of data. And apparently it's more data than the Modus website can handle. So I had to use program R to run each of my tags data sets sort of individually, and that enabled me to get the data from every single ping that the tags ever sent across their lives. So after playing around with program R, and I'll get into that in my next slide, I believe. So after using program R to run each tag, I was able to get thousands and thousands and thousands of data points, each one corresponding to all of the pings that the tag ever sent. And all this data was in the form of an Excel spreadsheet. So I ended up with 5 giant Excel spreadsheets, each one listing the data associated with every ping each of the 5 tags ever sent, which was a lot because it was 38 seconds. Because it was every 30 seconds. And the tags lasted, you know, about 28 days. So program R. This was very tricky to figure out. I was like 1 iota above what is coding before I became involved with this project. So some special coding packages were required to obtain the tag data. And I was just like, at this point, I was like, I don't know how to do this. Like, I don't even know where to begin to know how to do this, how to start using program R to get my tags data. So I began emailing with Dr. Amber Roth, and I was saying, like, you know, do you have any experience using program R? I'm not really sure how to use it. And Dr. Roth basically said, I don't have experience with this, but I can hook you up with someone who does. So Dr. Roth introduced me to Emily Filiberti. I think she's here somewhere. And she's a grad student at UMaine as well. And she was able to help me basically learn the ropes of program R. And she was able to help me ultimately get those 5 giant Excel data sheets that I needed for this project. She was able to help me download the right packages that I needed to be able to see all of my data. So after the appropriate packages were downloaded into R with Emily's help, the program was able to access all of the invisible data from all 5 tags from the Modus website. So while not all of the data from each of the 5 tags was visible on the Modus website, all of it was there. It just wasn't able to be seen by the website just because there was so much of it. Like, it wasn't able to be shown on the website because there was so many data points. So program R was what was needed to sort of be able to see literally every single data point. So each Excel sheet for each tag contains about 16,000 data points, give or take a few hundred. So that's a lot more than I guess the Modus website was able to handle. But I now have I now have these 5 enormous Excel sheets saved in my folders that I was able to achieve through Program R. And one of the pieces of data that is associated with every ping that the tag ever sent was the location of the tag when it sent out that ping. So the so the location of each ping is also recorded by R on each spreadsheet. So there, all of the only location ever, all the tags were only ever picked up by the tower at the Fields Pond Audubon Center. It would have been really cool to see, the tags, being detected in some other part of the state or some other states even, but that never happened. There were a couple outlier pings from Montana, but there were just like 3 or 4 of those amongst the 16,000. So those were dismissed as noise or like coding errors. So these are the results of my experiment. So basically, to sum it up, it took an adult monarch an average of 4.6 days to leave the area where they lived their previous life stages. But one thing to note is that this average is skewed by one significant outlier. 3 of the monarchs, as you can see in this table below, you got the tag ID numbers on the far left here. Each one of these just corresponds to each of the 5 tags that I had. So 3 of the 5 monarchs stuck around or were detected by the Fields Pond Modus Tower for 3 days. And then one of them only seemed to stick around for 2 days. And then one of them seemed to stick around for 12 days. So that 12 skewed up this average. In reality, it's probably something closer to 3 or 2, but this outlier skewed up the average here. So let's talk about the results a little bit. So in general, monarchs usually remain localized for only about 2 or 3 days before moving on, if you were to ignore that outlier. But more research is needed to determine if the outlier is in fact an outlier. Because my data set was so small, I only had 5 tags. I was only keeping track of 5 monarch butterflies. So because my sample size was so small, it's tough to really make a call whether or not the outlier was in fact an outlier. Maybe if I were to do this all again, I'd find that, maybe lots of monarchs stick around in the area where they're born for, 2 weeks or more. I don't know. So more research is needed to determine if that outlier was in fact an outlier, and a larger sample size would help to improve the accuracy of the results. So like I mentioned before, all of the monarchs were only ever detected by the Fields Pond Modus antenna. So a couple possible reasons for this is that they died before they could reach the proximity of another antenna. Or another possibility is that just the battery died before any of the monarchs were able to reach another antenna. Or it's possible that the monarchs simply just kept out of range of the antennas. So the range of a Modus antenna is about 8 miles. And, so there are significant gaps between the towers across the state and across the country. So it's definitely a possibility that the monarchs just sort of flew out of range of all other antennas as they moved across the state and across the country. So what could be done? What could I have done better about this experiment? And like, if I were to do this again, what would I improve? So again, I sort of talked about this a little bit already. The outlier, the outlier sort of skewed the results. So really a larger sample size would provide more accurate results. And and that would help to explain the outlier a little bit better. And here are basically the same reasons restated for why the monarchs were only ever detected by the Fields Pond Modus Tower. These are all just hypothetical scenarios only. I don't know what happened, and I probably will never know what happened. But these are just possibilities of what could have happened, why they weren't ever detected by another tower. And again, like if I had a larger sample size, then the chances of any one of them being picked up by a Modus tower other than the one at Fields Pond, those chances would go up. So, I think the main thing that would make this experiment better in the future would be to have a larger sample size. So with that, that's all I had. So I just wanted to say thank you to all the people and some people who have been with me on this and helping me through this. I wanted to thank the Entomological Society and the University of Maine for scholarship funding. I want to thank Dr. Amber Roth for directing this research project. I want to thank Emily Filiberti for helping me with Program R. I want to thank David Lehman too for supporting the project and for hosting the Modus antenna receiver, and for all the help that he gave me when I was actually at the Fields Pond Audubon Center. And I want to thank the Fields Pond Audubon Center itself for being the physical epicenter of my research. And I want to thank Charlene Donahue and Dana Michaud for supporting me over the years in my entomological endeavors. You know, they really helped to fan the flames that were my passion for insects in particular. So anyway, this is all that I prepared. So sorry if it was a little bit shorter than what you guys are used to, but that's what I had. So I'll open it up to Q&A now if anyone has any questions or comments or anything like that.
Yeah, that was wonderful. Thanks so much, Wes. We do have a couple questions in the chat box. So yeah, you covered this in your presentation, but you might as well go over it again in case anybody missed it. You know, but a couple questions about the physical attachment of the antenna. So first up, does the antenna for the tag sit flush to the wing? If it does not, how does it not impede the monarch's flight?
Yes, that's a good question. Thank you. So basically, so the tag is glued to the underside of the abdomen, and the wings are connected to the thorax of the butterfly. They're not actually, the wings don't originate from the abdomen of the butterfly. So when the wings flap up and down, they don't actually ever come in contact or come near sort of the underside of the abdomen of this animal. And so this long, thin antenna that runs off the back never actually really touches the wings. You know, I suppose it may brush against it as the butterfly like were to sort of fold its wings above its back like they do. It's possible it would brush against it. But yeah, no, this natural flight was never impeded. You know, like I said earlier, like 3 of them just flew right away. You know, and their flight pattern, like to the naked eye, seemed indistinguishable from just a untagged monarch. So yeah, we figured this was the best position, like that would like for the tag to be, that would sort of cause the least amount of possible disturbance to the animal's natural movements and habits.
Yeah, right on. I think a lot of people might have imagined it would be dorsally mounted originally, but yeah, where you guys did it makes sense. So next up, Charlene asks, has this been done with monarchs elsewhere? And in addition to monarchs, has it been done with other insects that you're aware of?
Okay, those are good questions. Yes and yes. So there's one other study that I'm aware of that involved tagging monarch butterflies. I believe they were even using similar technology to this one. There were some people over in New Hampshire. I believe they were affiliated with a university over there. I'm not going to give any names because I'm not sure it's accurate, but I'm pretty sure there was a university in New Hampshire that conducted a similar project to this. Yeah, just basically just tagging monarchs instead of seeing where they went. That was sort of their main focus. And yeah, these tags have been used on other insects. It was tags similar to these actually that were used to find the first nest of Asian giant hornets in the US. An Asian giant hornet was affixed with a tag very much like this one, a little white pill with a long antenna coming off of it. That actually led scientists to the first nest found in Washington, I believe it was. So yeah.
I wouldn't want to retrieve that tag.
No.
Great. Next up, Bob asks another question about the antenna and whether or not it would interfere with the mating process.
Would it interfere with the mating process? That is a good question, and I suspect it would interfere with the mating process. Yeah, I didn't really think of that, but yes, I imagine it would. I imagine it would, because the abdomen is the part of the insect's body that houses the, you know, the reproductive organs, and that all goes on at the tip of the abdomen. So yeah, I imagine it would. So one more thing that I will add about that is that we timed this project thinking that the monarchs that we tag are likely to be the ones that are going to want to migrate south. So these ones might not have like reproduction in mind. They might just be trying to get as much distance as they can, but I'm not sure. I could be wrong about that, so I'm not going to say too much more about that.
That's a hard one to know.
Yeah, it is. Yeah.
All right. Okay, here's this question again, which we already answered. So Hanson, saw you re-asked your question about the antenna interfering with flight. So hopefully you did hear that answer again. Next question after that is, how much does an antenna cost?
How much does an antenna cost? That's a good question. I'm not sure. I wasn't involved with the process of actually setting up the antenna at the Fields Pond Audubon Center. That had already been established before I was involved with any of this, before I was involved with this project, you know, because it was going on and being used by some people over who were running projects on birds. So I wasn't involved with setting that up. I'm not sure how much it costs. So
Yeah, probably a bit. All right, next
yeah,
question
I
here.
imagine it would be.
Yeah, next question here from Margie. Seems like the Motus antennae are few and far between along the coast of Maine, where monarchs are likely to fly as they migrate down south in the fall. Do you think that could explain the lack of signals?
Yep, I feel like it definitely could. Yeah, I sort of touched on it, I think, once a little bit earlier. So something along the lines of like, yeah, it's like these tags don't, I'm sorry, these antennas don't have a massive range. So it would be very reasonable to imagine that a monarch just flew, as it flew south, just managed to avoid another Motus tower. You know, yeah, that's a very reasonable assumption that it just missed them all. So.
Sure. And then quick follow-up to that. I don't know much about this Motus network. Is there plans to increase those number of towers? Is this a growing program, so to speak?
I'm not sure. Yeah, I'm not sure. I believe it is. I want to say tentatively yes, but I can't say with certainty. So
Sure. Next question from Kathy. Did you track male versus female individuals in your project?
yes, I did. I didn't really notice any trends like male versus female. In general, I think there were 3 males and 2 females that I ended up tagging. And I didn't really notice any sort of major differences between the two. Like both of them were able to carry the tags, and both of them were able to just fly away with the tags and have no problem. So yes, male and females were both used in the study, but I didn't really notice any dramatic differences between the two.
Another antenna question, of course. What are your thoughts on whether or not the structure of the antenna might lead to additional pressures for predation from birds, for example, you know, with an extra piece of material to grab onto or so?
Yeah, I suppose it's possible that the presence of the tag could make the monarch more noticeable. I mean, like just looking at it right here on this picture that is up right here of the monarch with the tag on it, like it just looks like a long thin hair. So it doesn't impede their flight, and it's not really super like obvious. It doesn't really like stand out like super conspicuously. I don't feel like. So I suppose maybe it could like, you know, make it more obvious to a bird with the string attached,
Yeah.
but
There's room for another study there.
Definitely.
Yeah, okay. Next message from Kathy, question. Are you aware of other research that might lend insight into how long monarchs tend to stay in their larval habitat before migrating? Were your findings that some adults hung around for 12 days surprising or not?
Yes, it was surprising, only because the other 4 all stuck around for only 3 days or less. So it was surprising to see that one stuck around for 12 days. But again, I'm sort of hesitant to sort of make any sort of definitive comment on the nature of the one that stuck around for 12 days, just because my sample size was so small. Like maybe if I were to do this with, you know, this is ridiculous. But if I did this like with 100 monarchs, you know, maybe like, you know, maybe sticking around for 12 days isn't that rare of a thing. So yeah, so short answer, that was surprising, but more research really will be needed to see if it really is that surprising.
Sure. Question from Joanne. What were the dates of monarch release?
What are the dates of monarch release? So they were not all released on the same day. They were all released in, I believe they all were within the month of August. Other than that, I couldn't tell you off the top of my head the exact dates of all 5. They're all written down in my journal, of course, but off the top of my head, they were all released in August. That's all I can give you.
Yeah. Okay.
Near the end of August.
Okay. Antenna question again. So Kathy Murray asks, any chance insects can hear the pings generated by the antennae to the towers?
No, the insects cannot hear the pings. So the pings are only actually audible when they're only audible when there is an antenna plugged into my laptop. When the FunCube dongle is plugged into my laptop, then that antenna can hear the sound and play it through the laptop speakers. Yeah, the tag is totally silent when it's just out in nature doing its thing. So yeah, no, there's no
Gotcha.
chance that insects or anything else can hear the pinging of the tags when they're outside.
Okay, next question. If only the largest specimens were used, do you think a more robust individual would behave differently than a more petite individual? And do males and females differ in size among monarchs?
Okay, so I think the second part of the second question will be a little bit quicker to answer, so I'll do that one first. Males and females do differ in size, but only by like a couple tenths of a gram. So like on average, the females are slightly smaller, but it's not a dramatic difference. And we were mostly picking robust individuals to tag, only not because we were thinking how they might differ in their behavior compared to a more petite individual, but only because like we didn't want to affix a tag to a small butterfly that wouldn't be able to handle the weight. So we picked the robust butterflies, not because of anything behavior related, but just because that we wanted to be sure that they were strong enough to carry this tag around. So.
Great. Okay, next question here. I am not sure if you covered this, but did you account for the possible termination of the study if the antennae broke off in way? Either the wire itself or somehow the attachment itself broke off in some way, and how was this possibility accounted for?
Yeah, so that definitely was a possibility. I mean, I didn't try, pulling a tag apart to see how strong they were, but the tag is the antenna was basically like a hair. It was similar to a hair when I was handling them all. So yeah, I guess it is definitely conceivable that I guess the tag itself broke. I think the monarch itself would have to come across something pretty violent, in order to sort of like actually break it in that way. But yeah, that was definitely a possibility. And maybe that's a reason why, none of the monarchs were picked up at other towers. Maybe the tag itself was destroyed in some way before it was able to reach another tower. So yeah, definitely a possibility.
Yeah. And I might have missed it while I was doing other background stuff here, but so you said that the towers themselves have an 8-mile radius where they can detect it. It's only hitting it if it's in that radius, right? It can't actually communicate coordinates within that 8-mile zone. So, okay, I thought so.
Correct.
So, okay, next question. What was the context of other monarchs that you observed at Fields Pond? For example, the number of monarchs, and when did their numbers diminish for fall migration?
Their numbers began diminishing for fall migration about the end of August, and that was when all of my tagged individuals were released. So that's sort of the short answer, but yeah, I feel like that's what I have to say. Yeah, I think that's right.
Very good. All right. Does anybody have any other questions to add to the chat box? And alternatively, if people are interested to, towards the end here, you can also unmute if you wish and ask Wes a question in person, so to speak. A follow-up question from Kathy. My question regarding ping was not about the ping that was audible to you. It was the frequency being sent out of the device that isn't audible to us. So
the question is not, can you hear the pinging?
And the insect
audibly?
detect the frequency that we would have no ability to detect.
Oh, okay. I understand. I understand. No, I don't think so. I haven't heard of insects being able to pick up like higher frequency sounds than humans. I mean, I suppose that could be true, but I just haven't heard that. So yeah, that could be true, and I just don't know it.
That remains undetermined.
Right.
Another question. How high do you how high do you think the quality of the data you gathered is based on your results?
I really think that the results would have been better if I had a larger sample size. I mean, even if I had done like 10 instead of 5, I really feel like the results would have been a little bit more concrete, a little bit more clear, so I think that would be the one thing that I would change, next if I were to do this again, would be just like to get a little bit more funding and buy a few more tags. So
That's always desirable. And
yeah.
then, yeah, follow up, do you plan to do this again?
Yes, I do. Fingers crossed. I'm hoping to apply for a cougar grant here at the university next summer to to do this again. So maybe I'll be able to get enough money for some more tags next time, more than 5.
And then another question, will you be having another webinar on it?
We'll see. I could. I guess it depends on how much you guys found this one.
Yeah, we'd always be interested in a follow-up if there's more research. And then, yeah, I haven't mentioned yet, but this webinar will eventually, recording of this webinar will eventually be available on the MES website in the near future. And then just a comment, it was enjoyable, and I'd love to see how the data compares to a larger study sample. So yeah,
Cool,
I think
thank
we
you.
would all be interested. And yeah, a lot of other positive comments here in the chat box about a great job done, Wes,
Thank
and
you.
very interesting stuff. So
Awesome.
does
Thanks,
anybody
everybody.
have any final questions then? All right, I'm not seeing any right now, so I'll do some closing remarks and then see if anything pops up. But yeah, that basically concludes our December webinar. So thanks again, everybody, for joining. We'll reconvene in January on the 13th. Brandon Wu will be our next presenter in the new year there. So not exactly sure what that topic will be quite yet, or at least I can't remember, but we have time. So keep an eye out for those announcements. And yeah, with that being said, I don't see any other comments or questions. So I'll bid everybody a good night and wish everybody happy holidays and a happy new year. And we'll see you down the road a ways. So thanks, everybody. Take care.
Thanks for coming, everybody.
Thanks a lot, Wes. It's a great talk.
Yes, thanks again, Wes, very much. We appreciate you giving us your time.
Yeah, thank you. Thank you.
Adios.